Okay, in this screencast, the first thing I'm going to touch on is the comparison of genome sizes. So we already know that the amount of DNA in prokaryotic organisms is less than that in eukaryotic organisms. And IB wants you to be able to make some comparisons between species and how much DNA they have in their genome. So first of all, the definition of genome is the whole of the genetic information of an organism. Okay, so that's the entire nucleotide base sequences for each of its DNA molecules. And different species are going to have a different size genome, but the size of the genome doesn't necessarily mean, it can indicate the complexity and the number of proteins that an organism can make, but sometimes it isn't totally indicative of that. As you can see in the differences here, the T2 phage is a virus, right? And so this is a million base pairs, right? So this only has like 180,000 base pairs, whereas E. coli has 5 million base pairs. And so you can see that the virus has way less base pairs than the bacteria. And the bacteria is more complex. It's a living organism, unlike the virus. And then as we start to get to something more complex, it's going to make a lot more proteins when you get to something like, a eukaryotic organism like Drosophila, which is a fruit fly. You've got 140 million base pairs and so on. So they've got so comparing that to, and then to humans and so on, that you can make some general assumptions about the larger the genome, maybe the more proteins and, the complexity. However, that's not 100% true at all cases. And just having a lot of DNA, even in humans, doesn't mean we're more complex than, say, a dog or something else. But you need to be able to compare the genome sizes in a T2 phage. You need to be able to know how much there is, so 180,000. In E. coli bacteria, 5 million. Drosophila, the fruit fly, so you can see 140 million. And then Homo sapiens, that's 3 billion. And then when you get to the Parasaponica, which is a woodland type plant, it's got 150 billion base pairs, so a lot more. But often that really large genome can actually make it really difficult for it to replicate. So it's not always an advantage to have so much more DNA. Okay, so take the time, get to know those numbers. I'm sorry that you kind of need to memorize them. Try to at least be in the ballpark as best you can. We've got some other numbers you're going to have to memorize later on in another screencast. Okay, so now let's look at eukaryotic chromosomes in the fact that it's important that you understand that if you're looking at, the entirety, here's all the chromosomes, say, in a human. So we've got 23 pairs of chromosomes, and these ones are laid out on what we call a karyogram. We're going to look at those in more detail in another screencast. But the genes that we have are found on, so the gene, that's one unit of heredity, right? That's what's going to code for a protein. Those are found in specific locations within the genome. Okay, on a specific chromosome. So we looked earlier at sickle cell anemia is found on chromosome 11, right? And we get 2 copies of that chromosome. So we get, 2 copies of that gene, essentially. There's other things like the protein that allows us to detect blue is found on, the color blue is found on chromosome number 7 and so on. So different genes are found in different locations. Now, when we talk about the fact that you get 2 copies of each of these genes, because we have these 23 pairs, which is a total of 46, right? Those pairs of chromosomes together that will have the same genes are called homologous chromosomes. They're also referred to as bivalent or tetrads, especially if they're doing crossing over. But homologous chromosomes carry the same sequence of genes. Okay, so if this was chromosome 11, maybe right here would be the gene for sickle cell anemia, and this chromosome would have, or it's for basically beta hemoglobin, this chromosome would have that gene here and the exact same loci, on the other homologous chromosome. So they need to be found in the same spot. So, but the thing is, though, is that homologous chromosomes carry the same sequence of genes, but not necessarily the same alleles for those genes. So maybe this one has the normal beta hemoglobin, and maybe this one then has the base substitution mutation that results in the sickle cell beta hemoglobin. So you might get 2 of the same copies. Your homologous chromosomes would then be, homozygous, Or you could get two different copies, and then your homologous chromosomes would be heterozygous, having two different alleles for those genes. Another thing that something I want to introduce to you is the terms autosome and sex chromosomes. So we've got 23 pairs of chromosomes, and the first 22 pairs are considered autosomal chromosomes. They have lots of genes on them, but they do not code for sex. So the last chromosomes are the sex chromosomes, and those will determine male or female within the sex chromosomes themselves. So for humans, XX is generally results in female, and XY will generally result in male. So if you're looking at one of these karyograms, you can see we've got 22 autosomes, pairs of autosomes, and then one pair of sex chromosomes.